Hello, ladies and gentlemen, and welcome to today's broadcast on electron fragmentation-based workflows for characterizing proteoforms with Agilent Q ToFs by Dr. Joseph Beckman, CEO of Emissions, Inc., and Dr. Rebecca Glaskin, LCMS Applications Scientist from Agilent. And joining us also will be Cody Schwarzer, a field system specialist with Agilent. Welcome to all three of you. I am Susie Valdez of LabRoots, and I will be moderating today's event. Welcome, everyone. We want to begin by encouraging you to engage with us. You can submit as many questions as you want at any time you want during the presentation. Simply type them into the Ask a Question box on the left and click Send. We'll answer as many questions as we have time for at the end of the presentation. And if you have any trouble seeing or hearing this presentation, just click on that support tab found at the top right or use that Ask a Question box on the left. Please join me now in welcoming all of our presenters. You may now begin your presentation. Welcome to all of you.
So today, Cody, myself, as well as Professor Beckman, are going to be talking about the practical electron-based fragmentation for characterizing proteins. First, I'd just like to show a comparison of CID and ECD. And first, I'm going to start off with traditional mass spectrometry, just doing the tandem mass spec of CID. Here you have collision-induced excitation, which you follow by unfolding and loss of labile groups. Then you get your bond dissociation and your fragment separation. But with CID, you can't get site localization of where your labile modification occurred. Electron capture dissociation starts by irradiating your biomolecule with low energy electrons. You get initial electron capture, followed by charge neutralization and fragmentation. Then you get your fragment separation, but you get able to retain your labile modification along the backbone of your peptide fragments. Now, if we just look at a comparison of the timescale and fragmentation nomenclature of tandem MS techniques, we see CID is typically on the microsecond activation timescale, while electron capture dissociation is about 10 to the minus 15, 10 to the minus 16 seconds. So it's a much faster activation time. When we look on the right, we can see the difference in fragmentation nomenclature. So for CID, you're typically getting your BY ions from cleavage of your amide backbone. And then for ECD, you're typically generating your CZ ion assignments. So here it was showing a schematic of the ECD cell generated from emission. And it's going to be incorporated into our Q ToF family. And here we see the ECD cell attached to a shortened collision cell that can be easily incorporated into our Q ToF, as shown here. For this ECD cell installed in the instrumentation, the tuning is straightforward using a swarm auto tuning that's included in Mass Hunter acquisition software. You can still able to have the same transmission as CID in resolution as what you had originally before installation of the ECD cell. The EXD controller tracks the operation of MS1 and MS2 operation of your instrument. And with this ECD cell installed, we have multiple software workflows to get better protein characterization, generating from bottom-up to top-down analysis. With this cell installed, you can also use it for coupling with LC on the front end of your system, as well as capillary electrophoresis and our ion mobility platform. So this is just showing how ECD cell works. You start with your +6 ion. You get charge-reduced species here in the yellow spot where your electron cloud is. Then you get your fragments generated out in the output of your collision cell. And with this EXD's major advantage over ETD is that with the ECD cell, both peptides and proteins can be activated to increase your side chain fragmentation. With ETD, you typically have to cool your protein molecules to mix with the anion ETD reagent. And this favors electron transfer, but not dissociation. So it reduces your side chain fragmentation. And one of the challenges with ECD is how do you confine these low energy electrons? These low energy electrons tend to strongly repel each other. Emission has figured this out by efficiently using magnetic fields to restrain your electrons into a tight beam to fragment your molecules passing through the electron cloud. So you can see the electron cloud here in yellow along this schematic picture. And you can see the flight path of your molecular ions. You have the ions going in, they enter the electron cloud, and you fragment the molecular ions, so therefore exit. How does this EXD work? Well, it uses magnetic fields to constrain your electrons radially, and it uses DC static fields to trap the electrons axially. And here we have the time scale for the EXD cell. It's typically on a microsecond time scale. So you can see EXD fragmentation is complete by the time the peptides or proteins fly through the EXD cell, about 5 microseconds. And you can see that it couples really nicely with these front-end separations such as ion mobility, capillary electrophoresis, or liquid chromatography, and can be easily incorporated into our time-of-flight platforms. So this is just showing some pictures of what's used for tuning of the ECD cell. So first, in this top upper left corner, we have the power supply that controls the ECD cell. The EXD controller is underneath it. This window shows that you can tune it with manual tuning, the voltage applied to the lens elements and the magnet, or you can use an auto-tune feature. That way, the EXD controller can auto-tune the cell for you. There's also a nice visual voltage graph in the upper right corner, allows you to look at the voltages applied to the ECD cell. In the bottom right, you see the filament parameters. This is where you control the current applied to the filament of the ECD cell. So now we're going to go into some data. So this is an example of some data we collected on the 6560 in collaboration with Kathy Costello's lab at Boston University. This is looking at the +3 ions of substance P. And here we have our mass spectrum on the upper plot, our drift spectrum on the left, and our abundance map in the middle. And we can see in the drift spectrum two structures, a compact structure and an elongated structure. And if we look at the corresponding mass spectrum for each of these conformers, you can see that by a CID, by applying 10 volts to the collision cell, we see our compact mass spectrum on top and an elongated mass spectrum on top. And we can see through the CID spectrum for each of these conformers, it's pretty identical. There's no distinguishing features between these two conformers. And we got pretty good ion assignments across the peptide backbone. Now if we formed ECD on those two same conformers of substance P, we have our compact ECD spectrum on top, elongated ECD spectrum on the bottom. And two things we can easily notice inside these highlighted red boxes are the relative abundances of the C-Z ion assignments. You can see the ratios of these C-Z ions in these red boxes vary depending on the compact versus elongated structures. And you can see in the upper right corner, we still got really good sequence coverage across the peptide backbone. Next, we wanted to look at the tryptic digest of NISMAB via CID and ECD. So look at site localization of glycopeptides to see if we can retain the glycans on these peptides. On the upper plot is showing the TIC trace of the NISMAB tryptic digest. The bottom is showing MS spectra of the following glycopeptides for GOF. And we can see in the MS spectrum, we see mainly composition loss of the sugar moieties from the glycan structure. Really don't see any pertinent information regarding the peptide backbone cleavages. Now, if you look at the same glycopeptide via ECD, you see a lot of extensive information within these plots. You see a lot of CZ ion assignments, as well as site localization of where the glycan is along the peptide backbone. And you can see we got good sequence coverage across the peptide backbone, as well as site localization of the glycan. We look at another example of the G1F glycopeptide, for the following glycopeptide shown here. We have our TIC on the top of the NISMAB tryptic digest, as well as the corresponding CID MSMS spectrum. And again, the CID MSMS spectrum shows mainly cleavage of the sugar moieties off the glycan structure attached to the peptide. We really don't see much information in regards to the peptide backbone cleavages. Now, if we perform the same analysis with ECD, we see a lot of sensitive information regarding the peptide backbone cleavage with the CZ and ion assignments, as well as site localization of the glycan moiety within the peptide backbone.
So we can see from Rebecca's example here that these spectra can really be quite complex, and we're going to be relying really heavily on our data analysis software in order to extract as much information as possible and really minimize that manual interpretation. Before we begin, my name is Cody Schwarzer, and I work with Rebecca and Emission to help develop some of the data analysis workflows that we'll be using for the EXD Cell. Now, today I'm going to be sharing just one of our options. This allows for our raw EXD data to be incorporated directly into your protein metrics pipelines. Protein metrics, or BIOS, as many of you may already be aware, is a vendor-neutral software. It's used by many of our customers already for day-to-day CQA analysis. And Agilent already has a relationship with protein metrics as we use their HCP tools as part of our own host cell protein workflow. What we're proposing here is that we also incorporate PTM to process our EXD data. Now, the workflow is fairly straightforward. The data is pre-processed using a tool called ExpProcess. This has been developed by Emission, and Joe will be going into some further detail later on about its specific capabilities. But in this case, we can do things like threshold, centroid, do some MS2 summing if that's what you wish to do. And then once we're finished, we'll export a .mzml or a summed .mzml file that can then just flow directly into our Bionic software. Fortunately, Bionic is a pretty easy to use interface, and we don't need any modification of the underlying software in order to start processing those EXD files. So I have a quick video here just to demonstrate how this would work if you're new to protein metrics. Now you'll see it's fairly straightforward. We'll select our MS data file and define our FASTA file. We can add things like decoys and common contaminants. Then we have our standard matching parameters, obviously in this case ECD and ETD. We can add in PTMs and glycans that we might be interested in looking for. Can also select some FDR cuts if we desire, and then go ahead and run that file. Now I have a pre-baked result here, and you can see this is BSA coverage. Over on the left, we have some nice sequence coverage map, and if I choose a particular peptide of interest, I can easily see all the matches and the mass error associated with those MS2 matches. Now you can see they also have a ladder and a pretty easy to find table, so you don't have to look that up. And now we can just zoom into individual ions to validate those matches. What I really want to emphasize here is this is an established software, and it's already a turnkey solution, and we're not really making any significant changes to your existing CID workflows. We don't have to modify anything significantly, and we probably more importantly don't have to retrain all of your end users on a new software package. And so with that, I'm going to hand it over to Joe Beckman. He's going to discuss some more advanced options and some kind of custom data analysis tools that we can use.
Thanks, Rebecca and Cody. We were really excited when we first got a hold of Agilent loaned us a high-resolution Q ToF to try our cell in. And after installing it, we were able to get beautiful spectra of ubiquitin as shown here. We were doing this from the 6+ charge state of ubiquitin, which is known to be tightly folded. And in a few seconds, we could collect a very complete spectrum that would give on the order of 95 to 97% coverage from both directions, as shown here. And you can see even in the enlargement on the bottom that the small peaks that are a very large mass are still nicely resolved in the Q ToF. But we quickly realized that we were going to be overwhelmed with information because it was taking us hours to hand annotate the spectrum. And so we started to look at different programs and different ways of analyzing it. And one of the things when we first put this into an analysis program that we discovered was that we were missing a couple of really important features. And these were side chain fragmentations that normally aren't annotated. But because of the enhanced ability to activate ions that Rebecca alluded to, You can see the loss of isopropyl groups from leucine and ethyl groups from lysine. So we're able to distinguish the isobaric leucine isoleucine couples by the side chain fragmentation. So that's important because 16% of the human proteome is naturally or consists of leucine and isoleucine. So it gives you even much more complete information than you would get from ETD and other fragmentation methods. So we've spent quite a bit of time in the past couple of years figuring out how to process the data that comes out of this. And as Cody mentioned, we've created a program called ExpProcess that will both annotate the spectrum as having been processed or collected with ECD enabled, and output it as some spectra in MZML files that could be read by third-party software. We've actually built this platform with the idea that we could provide much more complete software, and we're using an open source set of programs that are known as KNIME, which is a data science open source program developed in Germany. And it's merged with OpenMS, which is also a huge open source project. And it's described here in this project or in this publication. So it's been under development for 17 years, and it has an enormous number of functions that are really practical for mass spec. And you can wire these together by just connecting through a graphical interface different modules and functions that you want to do. So this figure 2 was pretty intimidating. We're using a very basic one as shown in the above, the figure above it. And we're using an open source program called MSGF+, but you can very simply change out MSGF+ to mascot. And if you wanted to get false discovery rates, you can insert percolator, which is a very common means of estimating false discovery rates. And then we wrote a program that's called ExpViewer to be able to look at the results and see what the spectra look like, because it's always nice to see what data were you getting. Now, the nice feature of KNIME is that you can group all these functions together, and we can provide workflows for lots of different applications. And you can create a library of these that are essentially transparent black boxes. So you don't have to be an expert, you don't have to know what's happening underneath, but as you use it, you can open it up, you can examine all these functions, and you can draw on a very flexible platform to add more and more features. So this can grow with you, and it's also something we're continuing to develop. Now, to be able to look at the data, we created ExpViewer, and this is displaying the data from ubiquitin that I showed you in the first plot. And what we've done here is generated a fragmentation map that also is annotated with the number of spectra that are shown. So if you start, the blue dots down here show you the C ions that are generated, and you can see they go all the way through with two notable gaps. And these are where prolines exist. And because of the proline side chain being cyclic, you don't get ECD fragmentation across prolines. And if you look on the top panel, you're looking at the Z ions coming from the other direction. Okay, so you can easily visualize the types of fragmentation that are occurring. There's also some CID peaks in there, and there's fragmentation that generates A ions that's also present in the spectrum. The other thing we did is we added tilde marks here to indicate that you have side chain fragmentation. Now, this is part of a program, or this program is in a set of features that lets you examine spectra. And I'm doing this with a middle down workflow. So one of the things that I forgot to mention is that with ECD, working with larger peptides and in fact entire proteins is actually better because electron capture increases with the square of charge. So long peptides and proteins fragment even better. We're showing this with lyc digests. And here's a digest with sulfidyls reduced. And we're generating fragments that are quite long, and normally you wouldn't be able to analyze with CID. And we're going to focus on the glycosidic peptide, which Rebecca was just showing you, being done with trypsin digests. Here we're working on much larger fragments. And the workflow is basically we run the digest, separated it, collected the data, run it through MSGF Plus to identify peptides, and now we're viewing the results. And that's shown here, where the chromatogram is shown above. I put a blue rectangle to show that this is where the N-glycoside peptide is eluting. This is the spectrum that's generated, and I've highlighted the peptide that contains the N-glycosylated peptide. So if you click here, this spectrum is annotated, and then above it, we can zoom and we can see with a ruler, here's where the glycan is found between two of the fragments. Let me go to the next slide and show you basically how this is working. So you can take your mouse and you can click on the different dots, and that will show the spectrum. So in this case, I'm going to start from the amino side, one residue before the N-glycan. And so you can see the C7 ion, the C8, the C9. Coming from the other side, you can see the spectrum as you walk in. So you're getting identifications with fragments on both sides of the chain. So you have a very high degree of confidence of what this is. There's also a leucine present 4 amino acids in from the carboxy terminus. And this is showing the spectrum that's seen here. So in this particular case, the side chain fragment is actually much more dominant than the main chain fragment, the Z4. And that has to do with the ions being activated and the side chain fragmentation coming apart very quickly after the electron capture event. So we're getting exceptional coverage of side chain fragmentations that really helps with identification. It's also very useful with the side chain fragmentation to note that that breaks cysteine bonds. And here's an example of analyzing insulin. So this is insulin that's not reduced, and it's eluting off a standard HPLC column. And you can see that you get very nice fragments with ECD spectrum that are nicely resolved. And if we start to analyze that, what we found was we could characterize where all 3 disulfide bonds were located. And to illustrate this, we have to analyze it in 6 runs, but basically we consider breaking this bond, we look for the B chain, and we consider the A chain as a post-translational modification. And we can get extensive fragmentation across the B chain, repeat the analysis, and you look for the A chain with the B chain is modified. And so we've got very strong evidence for this disulfide being present. And you repeat this process, but we then find evidence for all 3 disulfides and how they were localized. This works on much larger proteins as well. And here we're taking a NIST antibody, and basically what we're doing is breaking the disulfide that holds the light chain with the heavy chain. You can now see the light chain coming off as a series of resolved residues, or resolved chains, that you can figure out the light mass. And then you can use the ECD peaks that are located down here to start to identify which light chain is paired with which heavy chain, or to start to study where drug antibody conjugates might be located. So in summary, we have a very practical method of doing electron fragmentation, and now we've developed a very flexible set of workflows to analyze the data that's coming. We get exceptional coverage with middle down and top down proteomics. We're able to characterize disulfide bonds, and ECD is ideal for measuring, for locating post-translational modifications, and the ability to do it middle down means you can get a lot more information about the co-location of phosphorylation events and other post-translational modifications. You can look for artifacts such as the formation of isoaspartate from aspartate, which I didn't show you today, but that works really well, and characterize, distinguish leucine from isoleucine, which are isobaric. And there are a lot of other capabilities that for analyzing glycans. So finally, we are very proud to be working with Agilent and their logo with Trusted Answers. What I hope you can see from today is with the fragmentation you can get using our ECD cell, that you can get really even superior answers, or your trusted answers are enhanced considerably because of the additional information that you can get pretty easily. So thank you, and we'll be happy to answer questions.
Thank you, Dr. Thank you, Dr. Beckman, Dr. Glaskin, and Mr. Schwarzer for that informative presentation. And we are now going to move to the Q&A portion of our webinar. And I want to encourage all the participants to please go ahead and ask questions by using that Ask a Question box. So it looks like we already have some great questions coming in from our audience members. Our first question: can you add the EXD cell to an existing Q-TOF?
So I'll go ahead and answer that. And the answer is yes, that we've designed this to be a replacement for the standard collision cell. It takes about an hour, not including pumping, venting, and pumping down time to install the cell, and then to bring the system back together. So in fact, we're selling this as an add-on, even with new systems.
Thank you so much. And our next question: can you still use CID with the EXD cell installed, and does it still behave like a standard Q-TOF?
And the answer is that yes, you can use it for standard CID spectra or fragmentation. It still behaves and gives what appears to be the same fragmentation patterns. And you can combine ECD with CID to get complementary information, or to help shake apart large molecules and get much better coverage than is possible in ETD or with CID alone.
Thank you so much. Our next question: is there a simple way to service or replace the ECD filament?
Right. So the way that we produce electrons is we have a hot filament that sits in the middle of the ECD cell, much like a filament that happens in old vacuum tubes. And we anticipate the lifetime is between 3 to 6 months. But to replace it now, you have to vent the instrument, but there's a trap door that you can open up, and you pull out the burnt out filament and plug in the new one, and then restart the system. So it takes just a few minutes to pull that off, and the vacuum usually comes back in a couple of hours at most. It's operational usually within an hour.
Thank you so much. I want to give each of you a little bit of an opportunity to provide some closing remarks for our audience members. And again, any questions not asked today will be answered via email from our presenters. We'll start with you, Dr. Beckman. Would you like to provide any closing remarks for our audience?
Yeah, so it's been a lot of fun working with Agilent over the years, and particularly Rebecca and Cody, as we start to turn the capabilities of the ECD cell into actual workflows and solve real problems. And we're really excited about the results we're getting and the extension that can happen. And we're looking forward to working more with Agilent to tackle more and more problems. I think we saw earlier in John Vilsted's presentation a really nice example of what was done at the University of Michigan for analyzing BSA with collision-induced unfolding, ion mobility, and ECD. And there are a lot more examples of being able to solve the unsolvable now.
Oops, I apologize. And I also want to offer you, Dr. Glaskin, an opportunity. Would you like to provide any closing remarks?
Yes, with the EXD cell installed in our Q-TOF platforms, it shows a nice complementary technique to traditional CID experiments for better characterization of your biomolecules.
Thank you so much. And Mr. Schwarzer, are you able to communicate with us? I think we weren't able to have him log on, and I'm so sorry to our audience members, but I know he is appreciative of everyone participating today. Again, I want to thank all three of you for your great presentation and for your participation. today. And I also want to give another opportunity for John, who's joined us today, and he wanted to offer a few extra comments to our participants. John, would you like to add a couple of comments?
I would, thank you. Actually, these workflows have been just terrific in seeing the development and the effort that's gone in from emission, working together with our collaborators, and especially with Rebecca and Cody. Now that these workflows are coming to fruition, Agilent is actually initiating a promotion in North America for end of calendar year. So contact your local mass spec product specialist, as there is a very aggressive promotion in place for adding an EXD to an Agilent Q ToF if you're looking at end of year situations. So this will be good through the calendar year. Thank you.
And thank you so much. And as a reminder, again, to our audience, any questions that were not answered today and those submitted during the on-demand period will be addressed via the email you provided at the time of registration. And I want to thank our speakers again for their time and for those of you who participated. And we hope all of you will check out the exhibit hall and enjoy the rest of the Agilent event today and tomorrow. Take care, stay healthy. Bye-bye for now.